The stage is yours. Thank you so much.
Well, thank you. Well, I will say thank you first to Hamilton University for hosting this event. Also, to have the opportunity here today to speak. Cannaflos, because I'm a member of Cannaflos, we have registered our cultivars in the CPPO, but it's our connection with them. So today, I want you to take a journey on cannabis tissue culture, advanced preservation and propagation for medicinal applications. But first, I would like to share a bit about myself. I'm an agronomist by profession. I've been working in tissue culture. I started in 2014. My PhD in plant biotechnology, working first in many different species, watermelon, melon, garlic, tomato, many different species, trying to implement regeneration protocols in those species. And in 2019, I started working in cannabis in the laboratory, trying with the same purpose, trying to implement regeneration in vitro protocol. So, but then I jumped to Cannaflos in 2019, and as a member of Cannaflos, I'm the head of research of Cannaflos Genetics, that is the subsidiary company of Cannaflos GmbH. First, a quick but important disclaimer that all the images that you are going to see today in this presentation has been taken by our personnel, by Cannaflos personnel. So, important to say that. Starting with the presentation, I would say why is important tissue culture in cannabis and why now. To understand why now is important tissue culture is because during the prohibition era, cannabis cultivation and also research were pushed underground. So during many years, no research, no cultivation. So, but end of the 20th century, and specifically from 2019 on, there was a research boom. I mean, many publications in different areas like pharmacology, genetics, and cultivation techniques. In other sectors, fruit trees, ornamentals, tissue culture is in fact very important. It's essential, I would say, because there are many. These in general, all these species has been propagated during the decades from seeds, cuttings, and or grafting. But nowadays, these also traditional methods have some limitations, or could be also because of modern agriculture requisites that in the end, in every single species crop, they are demanding high yields, genetic uniformity, and disease plant resistance. So to overcome all these, tissue culture offers a promising complement to traditional breeding methods by providing control environment, control conditions for plant regeneration and propagation. Maybe most of you already know what tissue culture is, because it has been a topic in the last years, has been a topic in many different conversations. But by definition, tissue culture is a collection of techniques that enable the cultivation of plant cells in arsenic conditions and the exploitation of their total potency, as well as their aptitude for genetic variation. Here I wanted also to show you a few pictures, how it looks, our cultivars, our growth chamber, our equipment plants. So in general, tissue culture stands in five essential pillars. The first four are the air quality control, optimized lighting, the plants should be grown in arsenic conditions, and also in a tailored nutrient media. And the fifth pillar is cell totipotency. because totipotency is the ability of a single plant cell to regenerate into a whole plant under optimal laboratory conditions. Here with this picture, I wanted to show, starting from the left, a cotyledon explant that is showing a primordia growth at the bottom, and how this primordia evolves into an organogenic cali with multiple shoots. And from these shoots, we can elongate them, and finally, we can root them. Through this totipotency, there are different pathways to work in tissue culture. I mean, the embryogenic pathway or the organogenic pathway. Starting with the embryogenic pathway, it involves the acalus phase. This is because in the culture media, there is a high ratio of auxin and cytokinins, but we have to take into account that with this strategy, there is a high rate of somaclonal variation. If you are looking for mutations, if you are looking for different phenols of cannabis, that will be a good strategy for you. But if what you're looking for is to propagate plants and to generate genetically identical clones, then you have to use the organogenic pathway that is faster and it doesn't involve a callus phase. And you can start with axillary bud or shoot tips. Also here some pictures, Petri dish on the left, Petri dish with cotyledon explants with a media containing high concentration of auxins. This is how it looks like the embryogenic cali. And next to it, also a Petri dish with full of cotyledons that are regenerating some plants. This is the organogenic pathway. You can see the different shoots, and next to it, a close-up of this shoot that is focused on the meristematic part. In tissue culture, there are many different techniques, starting with, for instance, somaclonal variation that I already mentioned, embryo rescue, synthetic seed production, but there are more. I mean, genetic engineering, also polyploid plant production. But today we are going to focus on these two, on micropropagation and germplasm preservation. Starting with micropropagation is one of the most valuable tissue culture techniques used to mass propagate plant material in a highly controlled environment using explants such as shoot tips, meristem, or nodal segments. I wanted also here to highlight some of its benefits because you can produce a large scale of genetically identical clones, saving up to 95% of the space you need for your operations. Also very important to mention that you can rejuvenate your plant material by reducing the endophytic microbiome through different subcultures, and the year-round production that is offering this technique, and also for sure the year plus preservation. But this is not so, I mean, in tissue culture, also in micropropagation, is not so easy. I mean, it seems easy so far, but it's not. It's not plug and play. So there are many different, it's influenced by different factors, starting with the genotypic influence, the primary factor, because as you maybe already know, cannabis is a recalcitrant species. That means that it doesn't work many times in vitro conditions. You have to optimize the protocol. You have to work a lot to get a proper response, an in vitro response. So it depends on the species, and also is genetic dependent, the genotype dependent. That means that even in every plant, inside the strain, every plant, the in vitro response is going to be totally different. Also important to mention the explantor and its physiological stage, because if you have the opportunity to take explants from young plant, it's going to be in general better. And depending on your approach, you have many different tissues that you can take, starting from embryos, meristem, leaves, seeds. I mean, depending on your approach, you can take different explants. Here also, a close-up of a meristem explant, and below a petri dish with some leaves. Also important, another factors that are in our culture media, like grow regulators and also the nutritional factors. The grow regulators or the hormones, important here because if our culture media is a cytokinin dominance, then it's going to promote shoot initiation and development. But otherwise, if it's, our culture media is an auxin dominance, in general, it's going to stimulate the root formation. Going to the nutritional factors in our culture media, important to mention and important to take into account the medium composition. Different gelling agents, also basal salt, for instance, MS, DKW, these are the most trendy ones. Important to mention the carbon source, also the different vitamins, amino acids that are available, and also many times it works adding some complex additive like activated charcoal. Important also to mention the incubation conditions because our in vitro response is going to depend also in these incubation conditions. Important here to highlight also the light intensity, the light spectrum, the temperature of the conditions, because in general, it works better around between 26, 28 degrees. Also, very important, the gaseous environment, because all this is to ensure a good transition, a good in vitro response, and in the end, a good transition to the ex vitro conditions. So when you are going to grow in the end these plants, and when you're going to move to ex vitro conditions, important also to mention that your acclimatization strategy is going to be very important. Here, an overview, with this slide, an overview of the different factors that I already mentioned. And also here, I would like to highlight that it's very important when you are optimizing a protocol, try to optimize it as a system, because otherwise you are going to fail. You have to work as a system. Because if you are not properly managing your protocols, it's quite easy in vitro that appear molds, yeast, and even many different bacteria. And you can ruin your entire batches. Here, starting also with, because in general micropropagation, we can differentiate 5 different stages, starting from selection, followed by initiation, multiplication, shoot elongation and in vitro routing, and acclimatization and ex vitro routing. Starting with the selection, of course, is very, very important, as I already mentioned. If you can take samples, explants from young plant that has been growing in a controlled environment, is going to minimize the pathogen exposure. And if it's also beneficial, a pretreatment with some kind of fungicides and insecticides. Next stage will be the initiation. In this is when we are going, in this stage, we are going to sterilize our explant, and we are going to place them in a specific culture media to monitor and evaluate the growth response. Then, after checking also that it's free from pathogens, next stage will be the multiplication. Then, during this stage, is going to take a few weeks. You are going, is grown in general in a culture media with a high concentration of cytokinins. So in this way, you are going to start to see the shoots appearing from the explants. And then you can elongate this shoots in the next stage. In general, is by decreasing the cytokinin concentration. Then you are going to elongate this shoots, and you have the possibility to add some auxins to the media. Then you can root it in vitro conditions. Otherwise, you have another option that is like You acclimatize directly the shoot to ex vitro conditions, and then you use your common strategy to root your plants, your cocoa peat, rogul, uh, plaques, so you can root it. But I would like to emphasize here a bit with the acclimatization, because it's quite difficult sometimes, because you have to decrease the humidity by increasing the light exposure. So many times what is happening is it's very likely to happen that is going to become dehydrated, the plant, so you are going to lose them. Also here, an overview how the different stages interact between them. And here I would like to highlight this yellow arrow that is connected the stage number, the 1, the initiation, and the 3, the elongation, because it's skipping the multiplication stage. I mean, this is possible, a good strategy, depending on your approach, because if skipping the multiplication, what you are going, you in the end, you are propagating, is a technique that is clonal propagation, because from one simple explant, you are going to get one simple plant. Then through stage 2 multiplication, you can get from one simple explant many different explants. So this is this is micropropagation, this technique of mass propagate the plant. At this point of this presentation, also I would like to ask you about how would you preserve your price genetics, your early genetics, because during my visits to nurseries, to production facilities, I have seen many times the same. They are keeping their elite genetics just having a few copies in the same room, in the same, or maybe in another room, but they have a risk to lose them because they are exposed to different pathogens, not well covered. So, and no matter if you are a researcher, if you are growing cannabis because you are producing cannabis, so because if you are a researcher, maybe you have many reasons to preserve your genetics. Maybe you are working in a genetic diversity program, maybe studying its cultural and historical importance, because what is important maybe for a researcher is that you have your GERPLAS bank always available, free from biotic and abiotic pressures. And otherwise, if you also you are producing cannabis, also very interesting for you to keep your genetics in vitro conditions, because you can keep it vigorous across the cultivation cycles, maintaining a large genetic library in a small space, always having access to them, safeguarding them also against pathogens, and to preventing them potential loss of elite genetics due to external factors. For instance, if you can lose your license temporarily, if you have a third party, a third lab that is preserving your genetics, you can keep them. In this sense, Cannaflos Genetics is authorized by Spanish Narcotic Agency, the EMPS. We have granted with an in vitro and ex vitro multiplication license to keep our genetics and to propagate them. And we have in our team 2 PhD specialized in tissue culture that with many experience of that, and also that we are performing an in-house HLVD testing during the different processes. In the pictures, this is the pictures how it looks like our lab in Valencia in Spain, and also here some of our partners. Following with our multiplication license that we are holding, we are offering genetic germplants genetic preservation, also a model renewal service, because we are working with, we are acclimatizing the material directly sourced from in vitro conditions, tissue culture, and certified that is free from whole blood and viroid. And with this final slide, I wanted to to tell you about our Cannaflos and Cannaflos Genetics journey into the medical cannabis, starting from the beginning, giving access to our CPVO registered genetics that we are preserving in our tissue culture, also in ex vitro conditions in both. And we are testing continuously against hoblet and viroid, and how we can supply through our licensed partners, giving the different starting material through mother plants, also cuttings, and we are supporting them also during the cultivation phase and during the commercialization, because our matrix Cannaflos GmbH is a GMP and a GDP certificate. So that's it. So thank you for your undivided attention, and please feel free to ask.
Thank you so much. Thank you so much. I remember when I worked for Google, they always said, no backup, no mercy. Yeah, and this counts for the IT, but should also count for your varieties. So I'm asking you now, do you have any questions regarding the tissue culture operations?
Yeah, thanks a lot. Dimi here again, CSC Captain Widi. Several questions, as I worked with in vitro propagation, also with soft fruit production. Several questions. Do hormones influence the pheno and the geno? Yeah. And is it a mutation or is it the complete reproduction of the mother plant?
Last part?
I mean, okay, do hormones influence the pheno and geno from the mother plant? If we use hormones, probably we have some difference.
You can alterate the pheno, but in the end, you are not going to change. Depending on the pathway, of course, you are going, because if you are working with hormones like auxins, and if you are using the embryogenic pathway, of course, you can alter the DNA, through different mutations. Also, because this is why I already mentioned the somaclonal variation. It's depending on,
So, and how many passages do you keep?
How many?
Passages? I mean, from the mother, or you propagate, then you cut again, then propagate, cut again. How many times you make this? We made like around seven times.
But you mean from ex vitro to in vitro conditions, or?
No, in vitro, when you when you make your cut bigger and then cut it again. In vitro. How many times you cut ahead? I mean,
we started from ex vitro conditions. Then when it's implemented in our in vitro, then it's regularly propagated. I don't know if I'm answering you, but it's like I don't follow you completely.
I mean, you have a cut on your picture,
yeah.
and you make several cuts from them.
Yeah, you can, yeah.
And then we propagated them and have it the same and cut them the same. Do you do like that?
No.
Okay.
No.
You just made from the base material the batch of cuts and then it's done. Okay. Then what do you work? Did you work with bioreactors? And it's kind of different technologies.
I have seen it.
Liquid.
I don't know how it works, but I didn't.
Yeah. Your opinion to the synthetic seeds?
Good question, huh?
Yeah.
The real synthetic seeds. The real synthetic seeds, because I mean, in the end, a real synthetic seed is when there is an embryo inside.
Yeah.
And right now, many. I mean, I'm not going to say fake because it's not true. But synthetic seeds, many publications and many, it's like they are just putting like a shoot tip in this seed, in this artificial seed, and this is.
And what would you put in?
To be honest, it depends.
Tisha? A part of tissue?
No, of
embryo,
not.
as you say, it's embryo.
Of course, it could be a embryo, and the original one could be an embryo regenerated from the embryogenic pathway. That in the end, it contains the shoot, the apical part, and also the root, directly to place it and to acclimatize them to grow directly. Because if you are putting directly a shoot tip, then the recipient should have also the ability to root them, and it's quite complicated, quite complicated. But of course, it's working. I don't know many companies that is working through these synthetic seeds, but yeah, it will work.
Yeah, thanks a lot.
So there's one question from the online audience. They can also put their questions in the chat, and it's about Hope latent virus. So are there new insights regarding cleaning infected plants through meristem propagation?
I mean, yeah, this is a recent topic. They are doing right now a lot of research on it, and even they are regenerating plants through meristems, and still infected. So maybe good strategy also to combine with thermotherapy could work, but of course in the end you have to test. Maybe of course you are going to clean some. It doesn't mean that it's going to be all regenerated plant is going to be clean, but yeah, it works.
So are there any more questions? There is a question. Yeah, please.
Thank you, Carlos, for the presentation. Very clear. I am curious about somoclonal variation in the context of preservation. What do you think about it?
Depends on the strategy again. I mean, somoclonal variation using micropropagation is very, very reduced. I mean, you are not going to get this somoclonal variation, these mutations. Of course, it depends on the strategy, it depends on your pathway, even the very important, the tissue, the explant, the original explant you take. Because taking the meristem, if you take a meristem very small and you induce a callus, then of course the probability of getting mutations or to induce mutation is going to be so high. So it depends on your strategy. We can talk about it, but many different options.
Thank you.
In the initiation stage, what concentration do you use for the 5 to 15 minutes?
For the 15 minutes?
Because you said in the initiation stage you soak it for 5 to 15 minutes for the surface cleaning.
It's depends a lot on the original or the mother plant in this case, because if so young, then you have to decrease. Maybe you have to go to the 5. In general, I use 10 minutes, but of course, if it's too young, you have to decrease. You have to play with time, also the concentration.
And like ballpark the concentration for the like an estimate of the concentration, what would it be?
It depends. I mean, there are many different hormones. That is, as I said, this recalcitrant species. You have to work many different cytokines in general. Of course, it's going to be around 0.5 milligram per liter, something like this. But many is like a genetic dependent fully. And
yeah, thank you.
no, you're welcome.
I had a question about the role of the active charcoal in the micropropagation phase.
I started using activated charcoal to try to decrease the plant, because in the end, one of the limitations of, at least here in cannabis, is vitrification. Then I started using activated charcoal to decrease this vitrification, and it doesn't work. And also because already mentioned the endophytic microbiome, that in the end, you can see when you place your explants in this gel, you are going to see bacteria growing. And I like to use activated charcoal to try to reduce also this contamination. It works a bit. I mean, I don't mention a lot activated charcoal because it's like for me it's not fully working in general, and right now I'm not using it a lot.
Thank you.
You're welcome.
Hi, thanks for the talk. This was very interesting. I have something to add from the orchid industry. When you see pallets full of the same orchids in the shops, this is all tissue culture in Thailand, I think. They have rooms full of bottles which get agitated and then transferred into greenhouses with 100,000 of genetically similar clones. And it's really working on a very large scale.
Yeah.
And it's also working on a very small scale. You can buy bags with MS medium online and try it out at home and play around and see how it works.
Yeah, for orchids, for sure. I mean, during my presentation, when I mentioned ornamentals, I was thinking on orchids because in the end, it's on the contrary on cannabis. It's not a recalcitrant species. The in vitro response is quite good. Also, I remember papaya that has been during my work back into the university. It was amazing how it works. You can regenerate, you can multiply very easy. And it's like you can see a papaya tree, it's like, oh, you know, but it was so easy to propagate them. And with orchids, it's really, really work. And regarding your second question, no more questions? Good.
So, thank you so much. If there is not another question, I would suggest we do our coffee break. So 15 minutes from now, we are even good in time. So thank you again for this nice presentation. It was a pleasure to listen. And now just grab a coffee, grab something. Yeah, give us a clap. And for the online audience, you can also get a coffee at home, maybe. I don't know. It depends on your setup. And we will be right back at, let me look on the